Hey biology students! In this lecture video we're going to talk about treatments for viral infections. Pictured here on the slide we see a picture of remdesivir. You may have been hearing about remdesivir in the news because it is an experimental drug that may be an effective antiviral against the coronavirus, SARS-CoV-2, that is currently causing a worldwide pandemic. So we'll talk about remdesivir at the end of this video. So first of all, I would like to mention that there is a non-specific immune response that is tailor-made to specifically help the body fight viral infections. I left it out of our immune system lectures when we covered the immune system because there's already so many things to talk about in the immune system that I ended up eventually deciding to cut the discussion about interferons and put it into the viral lecture because it's most relevant as we discuss viral infections. And also, you really need to have an understanding of how viruses replicate. And if you've been watching these viral videos in order, you're now on the 5th video, you should have a pretty good understanding by now how viruses replicate using the lytic cycle. And in order to understand how antiviral medications work and how the immune system also helps to fight a viral infection, you really need to understand more about the life cycle. So now that you do, let's talk about the natural response that the body uses to fight viruses. So this response is called interferon. Okay, so let's tell the story of interferon. Now, once again, these are proteins that we consider to be part of the second line of defense. Remember, that is innate immunity. So it's part of the second line of defense, which means this is also a component of what we call innate immunity. So remember, innate immunity means innate immunity means a one-size-fits-all approach. So the immune system uses sort of a generalized approach to fight a pathogen without the targeted approach that, say, cytotoxic T cells or antibodies would provide in the third line of defense. So now that we understand how a viral infection occurs, we can understand how the body uses a protein called interferon to interfere with the production of new virus. That's where it got its name, by the way. Interferon has that word interfere in it, doesn't it? So it's the body's natural way of trying to block the effects of a virus. So what happens? Well, here's our virus in blue, little spiky virus, and it invades this target cell and begins to replicate inside of the cell, lytic cycle. Now, this cell knows it's infected. And what it will do as a result of the viral infection, it will produce these little proteins shown in red. And we call these interferon proteins. It'll produce it and it'll secrete it. So notice the proteins are coming out. Now we classify interferon into generally 3 categories. And when we abbreviate them, we write them as capital I, capital F, capital N. Okay, so those are interferon molecules being secreted by a virally infected cell. Now, a virally infected cell is doomed. Hopefully you understand that by now. So if a virus invades a cell, it's hijacking that cell. It is a doomed cell. It is going to be converted into a viral cloning factory, and that is it. Okay, so that is not a healthy cell, and that cell will die as a result of the lytic infection. And the interferon response, however, is designed to interfere with viral infection, but in these three ways that you see on the slide. So specifically, interferon alpha here will then travel to a neighboring cell and will bind to a very specific host receptor on this uninfected cell. Okay, so notice this is an uninfected cell, a neighboring uninfected cell. And it does this to warn, essentially it's warning the cell. And what the cell will do in response to the binding of interferon alpha to its receptor, this cell will then secrete a protein we call AVP. Okay, so they are going to secrete what are called AVPs. And AVPs stand for antiviral proteins. Okay, so it's going to make antiviral proteins. Let's give them another color so we're not confused here. So let's make these green. Okay, just so you're clear, interferon proteins are different than antiviral proteins. Okay, so interferon proteins are secreted by a virally infected cell as signals for these three outcomes here. Antiviral proteins are produced in response to interferon signaling and produced by an uninfected cell to protect itself from a viral infection. So what it'll do is it'll make antiviral proteins. And what happens is that if any virus happens to invade this cell, it will destroy the viral nucleic acid. So let's say now this virus comes knocking at the door here, and he gets inside. And remember, when viruses get inside of cells, they uncoat and release their viral nucleic acid, which may be RNA or maybe DNA. Eventually, though, viral messenger RNA is produced inside of the cytoplasm of the cell and used to direct the production of viral proteins. So the action of the antiviral proteins would be to recognize any RNA and destroy it. So put a big X there over the viral mRNA and show that it gets destroyed. So this puts the cell on high alert and allows it to destroy any viral nucleic acid in the cell. The second outcome of interferon is facilitated by another form of interferon. This one is called interferon beta. And so here we see interferon beta, the little red dot here, binding to a cell receptor. And what it'll do is it'll signal neighboring infected cells to undergo a process called apoptosis. And that is essentially a process we call cell suicide. So this interferon beta will tell an infected cell to commit suicide, apoptosis. I always remember that because apoptosis has the word pop in it. It makes me think about a cell bursting and dying. Why do we want a virally infected cell to burst or die? Because a virally infected cell is harboring new virus that's replicating. and it's no good to anybody. Okay, that cell is now hijacked by virus and is a viral cloning factory. That is not good. That cell needs to die before new viruses are born and released out of the cell. So the idea of interferon beta is to signal the cell to prematurely die in an effort to prevent the replication of virus inside of that infected cell. Okay, so that's option 2. See that? There's 3 options. There's option 1, there's option 2, all mediated by different forms of interferon. And then there's option 3. Here's option 3 down here. Option 3 is mediated by interferon gamma. Interferon gamma binds to immune cells, like macrophages, and stimulates those cells to initiate phagocytosis, okay, and begin engulfing the invading virus faster and more efficiently than normally. So it's an immune boosting function as well. So all of that sounds really good, doesn't it? Yay, immune system. Yay, natural responses. Yeah, we love natural responses. Sure, the immune system is amazing. Um, it's completely got my utmost respect. But there's some things to think about in regards to a response caused by interferon. Okay, so first of all, yes. Once people realized that how interferons worked, the first thing everybody thought of in science was, oh my goodness, wow, we make these proteins and they are effectively stopping the replication of the virus and boosting the immune response to the virus. Oh my goodness, let's bottle it. Let's manufacture it by recombinant DNA technology. Let's make a whole bunch of this, bottle it up, and just inject this into our virally infected patients. And voila, we will have been treating them with this natural chemical they secrete anyways, but we're going to give them a little bit more, and we're going to just clear this infection and pat ourselves on the back for being oh so smart scientists and realizing that there is this natural chemical that we can use to fight a virus. And this was initially done and used to treat hepatitis B and hepatitis C disease caused by the hepatitis B virus and the hepatitis C virus. And you know what? It worked great. And everybody did pat themselves on the back and say, yay, now we've got a really cool antiviral, and it's a natural chemical we make anyways, and we can sort of treat, we can give patients extra doses of this by injection, and we can just overstimulate this interferon response and clear the infection. And you know what? It did work really well. So further research helped us understand that there actually are some problems with interferon treatment. 1, some viruses have developed a resistance to antiviral proteins. So rather than the antiviral proteins effectively destroying viral nucleic acid, the virus has a way of destroying the antiviral proteins with an enzyme. And many cold viruses actually do have resistance to antiviral proteins. Later research helped us understand as scientists that amazingly, not all viruses stimulate an interferon response. So when a virus invades a cell, not all viruses will cause that cell to make the interferon signals that are necessary to signal other cells or signal the immune response. We also learned that interferons are short-lived. A lot of times they only last a few weeks. And if we have a virus that causes a chronic infection or a long-term infection, interferon response may not be an effective solution for long-term infections. We found that when we tried treating viral infections, with giving injections of this recombinant interferon that some patients had side effects to over giving them interferon, including nausea, vomiting, and fever. Okay, so those are some of the problems, sort of the pros and the cons of interferon. It is effective against certain viruses like hepatitis B and hepatitis C, but seems to have limited effect on other viruses with side effects and short-lived. So now let's discuss pharmaceutical applications to treating viral infections. And at this point, I'd like to probe you with this question. What do you think is easier to treat as a clinician? A viral infection or a bacterial infection? So based on your understanding now, your advanced understanding, if you've been following along with all the videos, which I'm sure you have, your advanced understanding of bacterial cells and bacterial cell structures versus viral particles and how they infect, you should understand that a virus, because it's an intracellular parasite, meaning it goes inside of the cells and causes an infection inside of the cells and is essentially not even a cell, that it is harder to treat than a bacterial infection in general. It's not across the board blanket statement, but sort of in general, because bacteria are prokaryotes and they have a unique cell structure with unique cell targets like peptidoglycan, or different sized ribosomes that we can use to generate antibiotics that effectively treat bacterial infections. That is, in general, considered an easier type of microbe to fight than a viral infection. And you know what? Many cases, probably most cases, when somebody comes down with a viral infection, they don't even seek pharmaceutical medication, prescription medication. Think about the last time you got the flu. Did you seek prescription medication? Did you even go into your doctor to get tested for influenza, which they can do with a nasal swab? Probably not. I've had flu several times in my life, and I've never gone to see the doctor or had myself tested or had any prescription medication. Flu took me out. It took me down. Don't get me wrong. I mean, I was, with the times I have the flu, I'm in bed for maybe a week or 10 days. I've had severe viral laryngitis. I've lost my voice several times, which is a terrible hazard of the job of being a teacher because your voice is sort of everything. And the times I've lost my voice has been really, really awful for trying to teach or figure that out. So, sure, viruses have taken me down plenty of times, but actually in my life, I've never actually used an antiviral pharmaceutical. And maybe you're similar. Remember, antibiotics are not effective against viruses. You know that. But there are some pharmaceutical medications that can be prescribed for certain viral infections. So we are going to discuss those briefly here. So we're going to call these pharmaceutical antivirals. So these are generated in a laboratory by scientists. They undergo clinical trials that involve first testing on human cell lines, then animals like mice or rats or monkeys, then eventually into humans, and then eventually approved by the FDA, licensed, and utilized by medical doctors. So these are used to prevent the spread and the replication of the virus, and they work by targeting specific stages of the viral lytic life cycle that you now understand. We'll talk about that on the next slide. I do want to mention that another approach to treating viral infections oftentimes involves pharmaceutical medications that decrease the immune response to the virus? Because the virus is definitely, if it's in a lytic stage, it's definitely causing direct damage to cells by infecting those cells and causing those cells to lyse and die. But also, viral infections are triggering an indirect response to the virus, which we consider to be the immune response. And we all know by now from studying the immune system is very powerful. And if it's overstimulated, which sometimes occurs, it can be such a powerful inflammatory response that the immune system is effectively causing all of the symptoms that you detect as a result of that infection. And it may even be the immune system in its fight, in its noble efforts to fight the virus. It's the immune system that actually kills the patient in the long run. So it may be important to treat our patients with immune suppressant medications. And even if you take Tylenol when you are sick with a virus, like if you have the flu and you take Tylenol, even I do that. I mean, I've never taken a prescription pharmaceutical for a virus infection, but I have many times taken Tylenol. Tylenol is a fever reducer. And that is what I mean by a medication that would decrease the immune response, okay? Like suppressing a fever or a cough suppressant, anything along those, a fever reducer like Tylenol, for example. There are prescription medications that can be prescribed that can further suppress the immune responses. But in this video, I would like to further discuss pharmaceutical antivirals, prescription medications that are designed to target specific stages in the life cycle of a lytic virus. So here's our general overview of the lytic cycle. And in general, pharmaceutical products that are designed to stop the spread of a viral infection utilize 4 general strategies. Okay, so there are 4 general strategies I would like you to know. And then in the future videos, we'll talk more specifically about these medications. Okay, so 4 general strategies. Now remember, the first stage of the lytic cycle is attachment. Okay, and so to prevent attachment, there are some antiviral medications that act as attachment inhibitors. Remember what goes into the attachment stage. The attachment stage involves a virus like this little enveloped virus binding to specific receptors on the surface of the cell. And remember, there's this viral protein here we call the ligand that must bind specifically with the cell receptor in order for that to allow the virus to enter the cell. And it's like the virus has a key to the cell, where the cell has a lock. The cell receptor is sort of like the lock. And the viral ligand, which in this picture would be this little spike protein, is sort of like the key. And that is the first stage to allow the virus to enter the cell, unlock the cell. So what happens is the antiviral medication acts as an attachment inhibitor. And so what happens is these drugs will actually block cell receptors. So imagine if there was a drug here. We'll make the drug yellow, okay? So imagine if this was the drug here. The drug would essentially plug up the lock. Sort of like if you have a lock and let's say you put a bunch of gum in it. So you gum up that lock with a whole bunch of sticky gum in there, and then you try to put the key in and it won't fit. The key won't fit in the lock. Okay, that's the effect of an attachment inhibitor. What happens next in the lytic cycle? Well, after attachment, the next stage is penetration, and then there's uncoating. Remember, in one of those mechanisms, what happens is the virus forms or enters the cell in what's called an endosome. And so it enters inside of the cell in a membrane enclosed vesicle that we call the endosome. So the virus would be inside there. And then the viral nucleic acid is released into the cytoplasm of the cell. So another type of drug approach are drugs that block endosome formation. And this also prevents not only endosome formation, but also the uncoating stage. So in other words, the viral nucleic acid is unable to be released into the cytoplasm. And these are drugs that effectively, they affect the pH of the endosome. And if you affect the pH, that can affect whether or not those proteins dissolve and release the viral nucleic acid. So that is the effect of the drugs that prevent the uncoating stage. All right, let's continue in the life cycle though. So after the penetration uncoating, remember then there's the synthesis stage in the life cycle of the virus. And in the synthesis stage, so in the synthesis stage, new virus is replicated. Simply, that's what's happening. So another approach of an antiviral is to prevent viral replication in various ways. In some cases, this involves inhibiting certain viral enzymes. Okay, and then the last thing that happens after synthesis, well, there's assembly, right? So synthesis, and then there's assembly, all the parts of the virus get together, and then the virus releases, stage 5, release. It's actually 6 stages, but I just realized I put penetration and uncoating in the same place. So hopefully that doesn't bother you too much. Anyway, so there is another target, and these are, so remember the virus has to lyse the cell and break out of it. So there are antivirals that prevent the release stage. Okay, so that gives you an idea of 4 general approaches to pharmaceutical antiviral drugs that might be prescribed for severe cases of a viral infection. Once again, that would involve attachment inhibitors, uncoating inhibitors, especially ones that block endosome formation or endosome function. There are antivirals that block viral replication in various ways, which one way might be inhibiting a viral enzyme. And then there are drugs that prevent the release stage of the virus, so it cannot effectively get out of the cell and go and invade any new cells. The last thing I'd like to tell you about is I'd like to tell you about how remdesivir works, the mechanism of action. So if you've been following the coronavirus pandemic at all, which I'm sure you have, you may be hearing about remdesivir in the news as an experimental drug to treat the current outbreak of coronavirus. Remdesivir is currently in phase 3 clinical trials, and it's looking to be effective. And prior to the human trials, remdesivir has also been tested in cell culture, as well as in mice models, as well as in monkey models. And it's shown to be an effective antiviral against coronavirus. How does it work? Well, remdesivir is an example of a type of antiviral that blocks viral replication stage. It blocks the synthesis stage. By now, you know that the coronavirus that is currently causing the COVID-19 pandemic is caused by SARS-CoV-2. So that is the virus we're discussing here. Now, I'm going to do an updated and super interesting and relevant and exciting video on coronavirus for next week's class. So stay tuned for that video. But as a sneak peek about that video, we're going to talk about remdesivir today. So we'll learn more about this virus very soon, but I'll go ahead and tell you right off the bat that SARS-CoV-2 is a single-stranded RNA virus, positive sense. So it's a positive sense, single-stranded RNA virus. And you know what that means, right? So that means that when the SARS virus gets into a host cell, we'll talk about all of its stages, okay? So just hang in there. But for now, let's just say the virus gets into its host cell, uncoats, and releases its single-stranded RNA into the cytoplasm of the cell. Let's make its single-stranded RNA look like this. So remember, in the synthesis stage, the virus will want to copy more of its single-stranded RNA. In addition, it will want to use the single-stranded RNA as messenger RNA to code for new viral proteins. So the effect of remdesivir. By the way, remdesivir needs to be given as an IV medication. So it's pictured here as a syringe because it wouldn't be needed to be given as IV. Anyway, so we would inject the remdesivir med into our patients, and that remdesivir would come into the cell. And what it will do is remdesivir is an example of a type of medication that we call, we're just going to call it R, capital R, okay? And this is the type of medication that acts as something we call a nucleoside analog. And the nucleoside analog is actually a way to think about it. It's a decoy nucleotide. Okay, so this single-stranded RNA will be trying to copy itself. It'll be trying to make more copies of itself. So it's going to want to be copying itself and making identical single-stranded RNA copies that can then be packaged into new coronavirus. Okay. And remember, that is facilitated by an enzyme. And that enzyme is RNA replicase. So what remdesivir does is it comes along and it actually looks a lot like adenosine. So we can, it looks like adenosine. Remember, there's the 4 chemical letters that make up RNA, which are A, U, C, and G. So that's the alphabet of RNA. So when RNA is being copied, that's essentially what we're talking about. We're talking about making a new set of RNA composed of the alphabet of letters. So what happens is this remdesivir will bind to single-stranded RNA. We'll just go ahead and bind it right here, okay? So this would be the remdesivir nucleoside analog, and it acts like a fake nucleotide, a decoy. Okay, so what happens is it binds to the single-stranded RNA, and then this prevents RNA replicase from actually copying the RNA. And so it prevents replication of the RNA, it prevents viral RNA from being copied inside of the cells. So that prevents coronavirus from being able to replicate inside of the cells and decreases this viral RNA and decreases the amount of viral particles that are produced inside the patient. And so that's why it's an exciting possibility as a treatment that could be effective in probably combination with other treatments to eliminate the SARS-CoV-2 virus from the body. Okay, so that is our discussion on viral treatments, and I will talk to you next week.